All right. Well, thanks for coming for my talk today. I know I was supposed to give this last year, um, and I got COVID and was really sick for 2 weeks, so wasn't able to. But yeah, I'm glad to be here today. And yeah, I'll be talking about some of my master's work, and my background in lichens and mosses. So I guess a little bit more about me. Like Tegan was saying, I grew up in Portland. I went to public high school and I was a part of the outdoor school program. When I was in sixth grade, at least every sixth grader for free got to go to outdoor school for a week in Oregon. And you got to spend a week there and do four different field studies: plants, animals, soil, and water. And then in high school, I ended up being on the soils crew and would go out and work with the sixth graders. And I feel like that kind of got me going in the natural history realm of things. And I ended up going to college at Wheaton. I played lacrosse there and did my environmental science degree. And when I was there, I started a farm on campus. And we didn't have Greek life, but we had themed houses. So I started the farmhouse, which is still going strong 10 years later, which is great. Um, and after graduating, I worked in Boston and gained my experience at the Harvard Herbarium and did a lot of flora projects and bioblitzes. And then I went to grad school at Eastern Washington University and worked with a lichenologist, Dr. Jessica Allen, who's pictured up there with me. And as of late, I've been here for the last 3 years being the curator of the herbarium. And I did get to present a similar presentation at the first ever Cliff Ecology conference last year. I didn't attend, but I did present virtually, which was great to be a part of. So cliff research is kind of up and coming, I would say, as we can gain access more to cliffs more easily. More research has been done, but I'll just kind of start off with a brief introduction on lichens versus bryophytes, right? So a lichen is a symbiotic organism between algal partner and a fungal partner, and sometimes as well as a cyanobacteria. And lichens are named after their fungal symbiont. Many lichens within the same groups have the same algal partner. Um, and they are often placed into different kind of morphological groups based on their growth forms, the main three being crustose, foliose, and fruticose. And today I'll be kind of talking a lot about those different groups. So I'll be kind of going back over and showing lots of pictures. And then as far as bryophytes go, right, bryophytes are comprised of three different taxonomic groups: liverworts, hornworts, and mosses. Today I'll mainly be talking about mosses, but I did find some liverworts up in the cliffs as well. And the main difference between a leafy liverwort and a moss is the arrangement of its leaves on its stem. I like to think of a moss as like the head of a pineapple, where the leaves are spiraling around, whereas on a liverwort, they're kind of in rows, side by side. So, lichens are not mosses. And I say this because unfortunately, a lot of lichens have common names with the word moss in them. So, I think a lot of people tend to get them confused. So, for example, here we have the Iceland moss, which is a lichen, Citraria icelandica, right? And then we have oak moss, Avernia prunastrii, a common one. Again, not a moss though, right? This is a lichen. Moss shingle, Seroma hypnorum, and lastly here, reindeer moss, Cladonia rangiferina, right? So even despite common names, right, make sure when you're looking at mosses and lichens that you're distinguishing them from one another. Lichen or bryophyte, a little quiz for us all. So this first picture up here, maybe you might think it's a fungi, but actually it's a basidio-lichen, right? So lichens can have structures where they grow out of their algal symbiont instead of kind of it being within it. Um, Next one here, have a true moss, right? Moss on a rock with a little sporophyte there. You can see we have a slime mold, right? Maybe it tricked you. Maybe you thought it might be a lichen. But myxomycetes are kind of their own unique group. A true fungi here, witch's butter, one that you might see in the woods, even around this time of year. a liverwort, a thalloid liverwort, Marcantia. Its fruiting bodies are kind of like these finger-like structures. And lastly here, just another lichen. So just kind of showing that lichens and bryophytes can be confusing because they can look like other things. And so um, hopefully maybe after today, after the presentation, you'll have a very clear picture of what they are in your brain. So why do we study lichens and bryophytes? Both are bioindicators and have been used for biomonitoring of air pollution for a very long time, whether it's by the Forest Service, which has like these long-term plots, especially in places like the West Cascades, where every year people go back and do monitoring of the air quality using lichens. They do epiphyte studies. They are both important microhabitats for many different organisms, whether you're a tardigrade or a mite, or maybe you're using a lichen or a moss as camouflage, like the Katie did on the bottom right of this screen. Um, Maybe you're using it to make your nest if you're a bird, right? And of course, there are some medicinal purposes, and you have to keep in mind here, right? The difference between a poison and a medicine is dosage, right? So be careful if you are going to use your usnic acid. You know, don't use a lot of it. Um, and you can find usnea in things like natural deodorant, toothpaste, things like that. Bryoria, the horsehair lichen, specifically the Spokane tribes, used to make kind of cakes and crackers out of it, especially in the wintertime as a food source when they couldn't really forage anything else. It wasn't the tastiest thing, but it got them through. And so there's lots of different purposes and ways you can study lichens and bryophytes. And why do people then study cliffs? So cliffs overall are really understudied, but they have a really unique diversity. Are hard to access, which is often why they are understudied. And they are also a really harsh environment. Um, you're just on a sheer cliff face. It can be hard to hang on. And they also have a really unique geological past, which makes them also interesting to study. On the lower right of the screen is a geological map of the cliff face of El Cap in Yosemite. So you can see all of the different rock types throughout it. And so different types of rock can also be intriguing depending on what kind of cliff face you're looking at, whether you're on the ocean, on an island, in the middle of the country like we are. Just keep it going. Okay. So some abiotic stressors of cliff ecosystems, we have slope, right? The angle of the cliff, rock heterogeneity, aspect, and insulation. So if you think about a cliff that has a high amount of canopy cover, right, it's going to be very shaded. Whereas if it's fully exposed, right, to the sun, it will be a lot different in terms of the species you might find there. And then, of course, water and light availability, right? If your cliff is along an ocean or a river, versus having no water source nearby. It might be very dry, it might be in a desert. Um, all of these different things make cliffs really unique. Rock heterogeneity, right? Meaning what types of features are on the rock? Do you have pockets? Do you have ledges? Those types of things, cracks. So cliffs do act as natural refuges and as old growth systems. There have been studies where they have found some Thuja occidentalis, oh, geez, up in the Niagara Escarpment. And they're over 1,000 years old up in these cliffs, right? So these systems are really old and they're often untouched, right? Because humans have not accessed them. So you can find endemic species to cliffs. For example, in Hawaii, over 50% of species that occur on cliffs are endemic to that one cliff, right? Um, there are often glacial relics in cliff systems because they can be high in elevation, right? So they might not have been hit by past glacial events. Um, and cliffs also allow species to escape competition and disturbance, right? So you can think of a crustose lichen high up on a cliff not much is going to disturb it, right? Um, and so just kind of some examples. Racemictrium microcarpum is a moss that is well known to cliffs and high elevation, as well as Umbilicarius nerifescens, kind of more known as a cliff lichen. But there's been quite a few studies on Thuja occidentalis up in these cliffs, and there have been quite a few studies on vascular plants, but not as many that have included lichens and bryophytes. So whenever they are included, we learn quite a bit about cliffs. Drones are kind of a new way that people are accessing cliffs these days for research, especially for vascular plants, since you can identify them through pictures. You can basically take a drone up, take a video or, aerial imagery of your cliff, and then you can basically identify a bunch of species and you can create quadrats and plot lines and transects and kind of do a study all through a drone, which is pretty cool. That way you can also access places that might not actually be physically accessible, right? So lichen and bryophyte cliff communities are often the most abundant and diverse taxa in most cliff habitats. They are understudied groups overall, and often when they are included in cliff research, new rare and endangered species are found. And in the arid west, the cliff flora is mostly unknown. For lichens and bryophytes, there have not been many studies. So, Bogus et al. 2017. This is a paper that was done in the Red River Gorge impacts of rock climbing study, and they found that in total cover in their meter squared plots. So this study included mosses, lichens, and vascular plants. So at the top here for total taxa, you can see Lepraria spa. So Lepraria is a dust lichen. You can see that it had the highest cover in the plots next to a rhododendron, and then the next three species were all lichens: Cladonia, Parmelia, Cladonia. And then you can see kind of the rank for vascular bryophytes and lichens overall and their total covers. And you can see that lichens kind of dominated with vascular plants kind of coming in second and then bryophytes coming in. So just kind of showing that it's really important to include not only looking at vascular plants and cliffs, but all of the different organism types. I'm hoping this one will play. Lichenologist Nathan Christmas, he is in the UK and does a lot of cliff work. He's a climber, and so he is able to use his climbing skills to access different cliffs. And I just kind of wanted to give you a little glimpse of the type of field work that is involved and the stratification of a cliffside and how as you go up the cliff, the species really change. Let's keep going. Okay, so as far as cliff disturbances go, we have more traditional disturbance versus human impact, right? So traditionally, cliff disturbances have mainly been from more larger fires or natural disasters, right? Not too many things can access a cliff. But then, right, we have humans, and with rock climbing kind of coming into play, and all of these different sports where you're going up into the alpine, mountain biking, camping, hiking, as well as, I guess, developing cliffs for housing, making mining cliffs for resources. All of these are impacts to cliffs. And um, And as of more recent, right, humans have been started to rock climbing. And so rock climbing has really, I think, come off strong in the last 5 to 10 years. And something specifically that they do when they go out to develop routes is they often have to clean the rock. So depending on your cliff, and how much plant material is on it, you might be scraping it fully clean. So the picture on the right of the screen is one of my sites from my master's project at Fisk State Park along the Spokane River. The cliffs there are covered in mosses and lichens, and in order to make safe rock climbing routes and to put in permanent bolts, they would strip the rock clean. And so when you go out there, you would literally see just stripes right, on the rock. It was pretty crazy. Um, and other times, right, you might be cleaning off rock that might fall, right? If you were to grab it, it would just get pulled off, right? So that's part of developing a rock climbing route as well. And some developers, they'll pick a cliff that's dry, right? So that every time they go back in the spring after winter, they don't have to clean it. And other people are more focused on the features on the rock and what is more interesting to climb, right? So they might say, oh, this has such a cool ledge, and then this crack goes up to this pocket, and like it's going to be such an interesting climb. But every single year, when you go there, you'll find a huge pile on the ground of moss because they have to clean it, right? So you have kind of different strategies or morals when it comes to climbers. And I guess also depending on where you are in the country, what rock type you have, etc. Um, you might find many different types of things going on. So with the rise of rock climbing in the United States, there are over 320,000 rock climbing routes. And this is coming from Mountain Project, which is a public site. And according from a paper from 2012, there are over 10 million climbers. So I'm sure that number is way higher now. When I made this PowerPoint last year, there were under 4,000 routes in Montana. So just in the last year, over 500 have been added, right? So this is the route development of new rock climbing routes. And when I did my study, I first grabbed my number from Mountain Project in 2019 and 2020. And so in the last 5+ years, and for Washington State, which is where I did my study, there have been over 4,000 routes added, right? And so a lot of the time people are going out and they're not asking permission. The park rangers at one of my sites, when I showed them the over 100 bolted routes, they had no idea they even existed, right? So a lot of the time this is done, people are going out, grabbing wire brushes and just kind of doing what they want. Other times they're buying land, right? And they're developing it themselves and keeping it private. Sometimes people withhold crags or information about where routes are. And so there are probably way more routes out there than what is public knowledge. Um, and as far as Washington State goes in the Pacific Northwest, the oldest climbing area in the Pacific Northwest is Smith Rock in Oregon. But as far as the Spokane area, in 1915, the Spokane Mountaineers Club started. And, that's when these guys were, hammering pitons into the rock, right? And scarring the rock. And so now that is not really a moral practice that's done anymore. They, drill into the rock and place permanent bolts. And of course, with, people like Alex Honnold doing all these crazy solos and documentaries like The Dawn Wall, or, climbing just got put into the Olympics as of 2020. It's just become a really popular sport. And where I went to for my master's at Eastern Washington, right outside of Spokane, there are over, I think, 6 rock climbing areas within 30 minutes of Spokane. And so there's a wide variety of granite and basalt to climb on. And yeah, it's just kind of crazy, the amount of routes there are to choose from at a certain point. Little background on rock climbing for those who don't know, right? So we have different types of rock climbing. You have top rope, sport, traditional, ice, and bouldering. Bouldering is not shown. You don't use a rope, right? So top rope and sport is kind of what I focused on for my study. Trad climbing is where you're placing metal pieces into the rock that expand. So you're not clipping into permanent bolts in the rock. Whereas if you're top roping or sport climbing, right, your rope is getting clipped into this bottom quick draw. So you would have your harness on, you'd have a ton of quick draws on you. And as you get up to your bolt, you take a quick draw, you put it into your bolt, and then you take your rope that's connected to you, right, and clip it into your quick draw. That way, if you fall, the last place you clipped into is where you would fall to, right? There are crags that have top rope access, which means you can climb to the top of the cliff and drop a rope down from the anchor. And then others you have to kind of climb up to get to the anchor. Um, and for route levels for rope climbing, they use the Yosemite decimal system. So you have routes with really large features that are easier to climb, or maybe the angle, right, is really low, so it's easier to get up the wall versus something that is more overhung, right, where you're kind of climbing upside down. So here's a quick map of just impacts of rock climbing studies that have happened. I wrote impacts of rock climbing review paper in 2021 with some friends. And so this is one of the kind of images we put together. And so you can see for the Pacific Northwest, there has been nothing done at all, right? Even though rock climbing, I would say here is kind of one of the biggest areas in the country. And so my study, right, was done in Eastern Washington. So a dot will be added there. But for the most part, they've been focused on vascular plants. And sometimes they have included lichens and mosses, but there have been no studies that have just focused really on lichens and bryophytes, especially out west. And I would say over here, they can be pretty dominant. So real quick, I just want people to know, so when you're doing studies with lichens, sometimes people will include them, but they'll only go to genus, right? So every species has a scientific name, a genus, and a specific epithet, right? And so if you just go to genus for lichens, you might not get the whole story, right? So here I just kind of wanted to point out, right, these two Xanthorias, really different between climbed and unclimbed plots, right? So if they had grouped all the Xanthoria together, their results would probably be inconclusive, right? So it's really important if you're including lichens or bryophytes in your study that you have a taxonomic expert or somebody who can verify or do your identifications, because a lot of the time in the past, they were grouping things into groups by genera, or even by morphotype, right? So they put all the crusts together and then they put all the foliose lichens together. And they weren't kind of looking at individual species. And so that's something that I wanted to do for my study. I kind of wanted to take it to the next level to kind of see how individual species were being impacted. So my objectives were to assess the impacts of rock climbing on biodiversity in Eastern Washington, to improve development practices and management of rock climbing areas for lichens and mosses, and to enhance lichen and bryophyte flora knowledge of Western North America. So for my study sites, I originally had 5, but I started my master's fall 2019, right before COVID. And so I had to kind of cut down my study quite a bit. The land in Washington was closed for over a year. Even if you had a government contract to collect data, you weren't allowed to go do field work. And so I had to wait. And then it was a bad fire year too, unfortunately. So I didn't get to do as much collecting as I wanted. But I had plenty of data, which ended up being great. And so I did want to include route age, since lichens especially grow really slowly. I was really intrigued. Like the older the route, do you have more lichens on it? Do you have more lichen cover on it? So I wanted to include that. So my 2 sites I chose, McClellan, which was in Fisk State Park along the Spokane River, the largest state park in Washington. Most of the routes there were developed by Eastern Washington University for their rock climbing program, their outdoor rec program. And so most of those routes were only 5 to 15 plus years old. Whereas my other site, Rocks of Sharon, which was the highest elevation site within Spokane city limits, uh, had some of the oldest routes in the area, some being over 70 years old. And these bolded numbers, so these were the numbers when I did my study, and these are the numbers now, right? So there have been a lot of climbs added to these sites since I did this research. And for each site for McClellan, I had 10 climb transects that I paired with unclimbed. And for Rocks of Sharon, I had 6. Abiotic factors I included were slope, aspect, rock heterogeneity, and canopy cover. And then my route variables were climbing frequency, climbed versus unclimbed, level of route, and age of route. Climbing frequency, I calculated based on Mountain Project and local guidebooks and the star rating of the route, how popular it was, um, as well as how long it took to hike to the route from the parking area. It had been found before that if it's easier to access and it's more popular to climb on, that more people will go there. Whereas if it's a 3 mile hike in, most likely it won't get climbed on as much. So I did my field work through August through December of 2020. All of my transects were 11 meters in height, and I placed my quadrats every 3 meters. My climb transects I scouted for and selected based on accessibility, age, and their popularity. And my unclimbed transects, I made sure that there was no visible climbing damage or mention of previous routes on the cliff face. And here, I don't know if you can see, but basically this is a drawing by Nancy Loomis from a local guidebook that I used. And so the black lines are your rock climbing routes, and the X's are bolts placed into the rock. And so you can kind of see, I tried my best to specifically choose my unclimbed transects so that they were not only next to the climbing routes, but they weren't too close to routes. So there would be like technically climbed areas on them, but also that the rock features along the route were similar to the unclimbed transect enough that they were actually comparable, right? Past studies, they did like a fully climbed cliff face and a fully unclimbed. And it's really hard to compare them if they don't have the exact same slope, canopy cover, rock heterogeneity, aspect, right? If they're not both north facing, if one's north and one's south, you're going to have a lot different species composition and makeup. And so they're not comparable. So I tried really hard to make it as easy as possible to compare my climbed and unclimbed transects. Okay. Survey methods, I had half-meter squared plots and I had placed them side by side 4 times, so 8 total plots per transect. I described or identified every species within each plot and I estimated their percent cover. I collected samples of each species in order to identify them under the microscope, since lichens and mosses are not really field identifiable most of the time. I recorded the slope of the center of each plot and every single rock feature I measured within each plot. And then I noted the canopy cover of the transect, as well as the aspect and then the height of each cliff face overall, and then of the plots. And rock features, right? Pockets, cracks, ledges. So I just wanted to make sure I was trying to get as much information as possible. Here's me doing some field work. Because it was during COVID, I would often only get 1 or 2 field assistants allowed to come out with me. I had planned to have like 2 huge field crews of 8 people and had all these climbers signed up to help me do the field work. And then I had to do all these COVID protocols. So often I'd bring a climber with me. They'd help me get my anchor set up. And then I would jug up the rope solo and do all my data collecting, and they'd just kind of hang out. But I had some guys help me, is Michael and Torin. So here's an example of kind of what a plot would look like. So every single species of lichen and moss within each plot, I would take a little sample of to bring back. And I would note their percent cover. So sometimes I would have 25 plus lichens in one plot. And I think the most mosses I had in one was maybe 5 or 6. For my identification methodology, of course, I mainly did morphological methods, right, using a compound and dissecting scopes to make cross sections and slides of specific features. And then, of course, for lichens, I had to take it a step further and do some chemical analysis. So I not only did spot tests, which you can see here, For example, you can use bleach on certain lichens, and as the color changes, it tells you what acid is within them. Lichens have specific chemical makeups for each individual species. So sometimes morphologically you cannot distinguish them, but chemically you can. And here is a TLC plate, thin layer chromatography. Each one of these lines is a different lichen species, and each color or length on this sheet here kind of tells you the different chemical components of that lichen. And so often I would have to do TLC for certain groups in order to get down to the species level or to be certain of what I was looking at. And so overall, I had 118 species of lichens that I placed into 83 groups for my statistical analysis. I had 27 species of mosses that I placed into 21 groups. I had 2 liverworts and 2 vascular plants ended up in my plots. Cliff fern and selaginella. Maybe if I had done this study in a different place, I would have had a lot more vascular plants show up. But that was kind of the unique thing about these cliffs, was that they were so, just abundantly covered in lichens and bryophytes. For my statistical approach, I won't really be going into my stats too much, but maybe for those who have a background or might know what some of these things are, it might be more intriguing for you. I did use mainly RStudio and the vegan packages and some ecological packages to do my analyses. I looked at richness and diversity by using raked abundance plots and linear regressions and Shannon's diversity index, as well as species accumulation curves. For climbing and site impact, I did an NMDS for all taxa and for just lichens, as well as I did a species richness ANOVA. And then among climbed transects by grade and age, site to site, I did an Adonis. And then for plot and transect abiotic variation, I estimated how abiotic variables contributed to richness and diversity through an Adonis again. And then I also did a linear regression. So here is just an effects plot of all taxa cover. So this is everything for climbed versus unclimbed. And I had a really significant p-value there, you can see. So overall, my study did find that that climbing did have impact on taxa cover. And so I kind of split up my results a little bit so that we could look at them a little differently. So if you look at individual sites versus me, pooling all of my data together, I kind of had different results. Since McClellan was along the Spokane River, it was mainly covered with mosses. So you can see that for bryophyte and plant cover, species richness, SR, and SD species diversity, that at McClellan, they were all negatively impacted. Whereas at Rocks of Sharon, the lichen cover was negatively impacted, as well as the lichen species diversity, but the mosses weren't, right? So then when I combined my data, I kind of had a different story. And so it was important for me to look at each individual site as well, because they were very different from one another. So how did climbing impact lichen morphology groups specifically? So I did split all of them up. Crustose, endolith, which means you only see the fruiting bodies on the rock, right? Lepros, your dust lichens. Fruticose, more 3D or pendant. Foliose, leafy with a clear upper and lower surface. Foliose umbilicate, it has a central holdfast that is holding it onto its substrate, and then foliosquamulose. And so you can see actually climbing had a positive impact on crustose lichens at both of my sites and as well as the crustose endolithic lichens at McClellan. And I think this was basically my hypothesis here was that crustose lichens, because they are embedded in the rock, they are able to withstand the stress of rock climbing, right? Whereas if you have this frilly, more 3D or leafy lichen, it's going to get scraped off right away, right? And so again, in places like McClellan, especially since they are literally stripping the rock clean of all of these mosses and leaving it bare, it literally allows the crustose lichens to come in, right, and colonize the rock. And with the other morpho group types, you can see it was a lot different, right? They had a very negative impact for the most part from rock climbing. So some common foliose lichens from my study. Parmelia saxatilis was in almost every plot, as well as Physia tenella, Physia fea, and Xanthoparmelia cumberlandia. The Xanthoparmelias are a group that you have to do TLC on. So I had, I think, two or three different Xanthoparmelias, and I could not distinguish them at all for the most part. So I had to do a lot of extra work on those. Whereas the Physia tenella and the Parmelia saxatilis, they were really field identifiable for me at a certain point. So it was really easy for me to throw those onto my data sheets. Common crustose lichens within my plots. The rhizocarpons were definitely common. I had two gray species, Disporum and Grande, and a couple different yellow rhizocarpon species as well, including Leccornorinum. Dimelanorhina showed up quite often in my plots, as well as Licidia atrobrenea, and Diploschistes muscorum also showed up. And that's a unique crustose lichen because it directly grows on top of mosses. Most common lichen genus overall in my study was Umbilicaria. I think I had 8 or 9 different Umbilicaria species in my plots. So here's just kind of a select few for you to see. These are the ones with that central holdfast that is holding them onto the rock. So they're kind of a unique group of lichens. I've seen a lot of Hyperborea and Phaya around town on rocks, even in front of people's houses. Whereas the Americana, I've seen bits here and there on exposed rocks in like up in Paddy Creek. Common mosses from my study, Grimmia trichophylla was in almost every plot. This is Centrichia ruralis. I don't know why the name didn't show up here. And then Hemalothecium fulgescens was also very abundant, and Antitrichia californica, which was a moss that I had not seen before since I did my undergrad and a lot of my work afterwards on the East Coast in New England. I wasn't as familiar with the mosses in the Pacific Northwest, but Antitrichia californica was a good one to get to know. Some cool species finds from my study. Henrica americana was a state record, and I only found it on climbed transects at McClellan, so it was only en route. The only other record of this species being collected was in 2002 by Bruce McCune here in Montana. So before that, it had only been known to exist in Europe. So this is the second record of it for the US, which was pretty cool. And yeah, it was really weird that it was only climbing on rock climbing routes. So I think Bruce was telling me that it really likes water. It likes to be by water. Where he found it in Montana, he had to take a boat across a lake to a cliff that wasn't accessible. And that's where it was growing directly on the rock. And so I think because Eastern went out there and they scraped the rock clean right next to the river, that this Henrica was able to kind of colonize and grow on the rock without any competition from the mosses. Henothecopsis subperoica, a pin lichen, was also a Washington state record, had not been collected in the state before. Sometimes when I was collecting in my plots, I didn't know, right, what I was collecting until I put it under the microscope because it was so small. So this one, I'm pretty sure I thought I was just collecting a dust lichen, right, a leprous lichen. And then later on, I put it under the microscope and saw the little pins, and I was like, oh my God, so cool. And that's kind of what happened with this Normandina pulchella too. It was a Spokane County record. It's really common west of the Cascades. It likes to grow on top of mosses. You can kind of see the moss in the background, but it's these little plates right here. And so again, I hadn't realized I had collected it until I had put it under the microscope. And then a new liverwort for Spokane County, Frillania californica. So, local management of rock climbing areas, what is included? It's often run by climbers. A lot of it is just seasonal cleaning, rebolting, right? So, eventually you want to put in new bolts so that you can still climb safely. Crag cleanups, a lot of leave no trace. And there have been seasonal route closures in specific areas for peregrine falcon nesting, for bats, but never really for plants. I know of one example of a really rare fern at a rock climbing site in New Hampshire where they ended up permanently closing one of the crags because that was one of the only sites that the fern existed. But for the most part, um, there have not been, it's not like people go out and do a study and they shut down a rock climbing area. And I think that's what a lot of climbers are afraid of happening. They're like, oh, we don't want you to come here. Like, we don't have anything special here. Um, but really, there are no really real regulations around route development. When I was at Eastern, I became a part of their rock climbing club. And at one point, we went to go bouldering in this place called Tum Tum. That was kind of a new area. And the president of the club had brought a huge bag of wire brushes and handed them out to everybody and said, go create your own boulder wherever you want. And I was kind of like, oh my God, that's crazy. Uh, so a lot of the time, right, species of concern are unknown because there is no preliminary list or information often about the site, especially for lichens and mosses or what species are existing up in the cliffs. Um, and some Pacific Northwest climbing organizations, a lot of these ended up funding my project as well. Locally in Spokane, you have the Bauer Climbing Coalition, Epic Adventures, Access Fund, Pacific Northwest, and then Mazama's Pacific Northwest. And so all of these organizations, they'll raise funds to like redo trails to crags or to go out and do a big cleanup. One of the areas in Spokane is like right in the city, and so they get a lot of graffiti at the crag, so they'll go like literally clean it off. And so yeah, but as far as kind of, oh, this is a new site, maybe we should look at what's here first before we develop it. That's not really something that people have thought about. So some management suggestions based on my study. I'd like to update development practices. You don't have to clean off the whole cliff face or even the whole strip that the route will be on. Stick to the holds along the route and any loose rock. Not every cliff face needs to be developed and not every single possible route needs to be created, right? There are some places that should be left untouched. And cliffs with high bryophyte foliose and fruticose lichen cover should be reconsidered for development, whereas cliffs with higher crustose lichen cover should be prioritized. Education and climber engagement, right? So we should be including climbers in these studies and in conservation management. We need to educate climbers on the importance of lichens and bryophytes, as well as create guidelines for seasonal cleaning and development to just restrict the amount of damage to these communities. And of course, biodiversity knowledge matters. Maybe we should think about doing surveys for not just lichens and bryophytes, but for all plant and animal life before we develop a new area for climbing. So just real quick, some acknowledgements. Um, a lot of my local rock climbing information I got from the Eastern Rock Wall, Epic Adventures, which was a recreation kind of club that took people out and did things on campus at Eastern. And John Shields, he was the lab tech for the biology department, and he was a retired climber. And so a lot of the old guides that he had physical copies of, I wouldn't have gotten if I didn't have them from him. So some of the oldest routes in my study were thanks to him. I did get grants from the Access Fund, the American Alkaline Club, the American Biological and Lichenological Society, the Society of Herbarium Curators, as well as the Northwest Ecological Research Institute to do my study. My field assistants, fellow cliff ecologists, Georgia and Laura, I wrote that review paper with, and they helped me a lot with my um, kind of study design, as well as Peter Clark, who is an environmental consultant that does cliff work, which is pretty cool. And then just my advisors and some extra taxonomists that helped me verify all of my specimens. And yeah, that's about it.